So let's talk about reproductive physiology. Welcome to our lecture. Here's our outline. We're going to talk about what determines sex in humans. How do we form gametes and roles of different hormones, feedback loops. You're going to love that. And then once we understand that, then we can start talking about physiological birth controls and how they work. So first of all, what determines sex in humans? Well, as mammals, our sex is determined by chromosomes, specifically the sex chromosomes. So we have how many chromosomes? 46, right? You get 23 from the egg and 23 from the sperm. They come together in fertilization and you have the beginning of a new human. So 22 of those chromosomal pairs don't determine your sex at all. They determine all kinds of other traits. And then there are this 23rd pair, the X and the Y chromosome, that depending on your combination is going to determine whether that fetus develops into a male or a female. Of course, there are some anomalies like triple X. Some women are XXX. There's also something called Klinefelter syndrome, which is XXY. Those are men, but they have an extra X chromosome. So there are some anomalies. So we'll recognize that. And if you have questions about those, we can certainly talk about that in class. However, for most people, the males are XY, females are XX. So which gamete is going to determine whether someone is a male or a female? The egg or the sperm? Yeah. So the sperm, right? So since females are XX, they can only make eggs with the X chromosome, so they're going to pass that on to whoever. But males can make two different types of sperm. They can make sperm that carry the X chromosome and sperm that carry the Y chromosome. So sperm with the X chromosome, when it combines with an egg, that's going to be a female. The Y chromosome, when that combines with an egg, that will become a male. But I knew when I was growing up, there was a family that had 5 boys, no girls. And then we also knew a family that had all girls. I can't remember, like 6 or 7 girls, and no boys. So what's going on in those cases? Well, it's not that the male is only producing X sperm or only producing Y sperm. Rather, they think what's going on there is the female reproductive system is somehow selecting for either all X chromosomes, allowing those to, X sperm carrying X chromosomes and allowing those to survive and culling out the Y chromosome sperm, or vice versa, that it's actually the female reproductive system that's doing some selection. Google that, that'll take you down a rabbit hole. All right, moving on. So this is an interesting sketch, kind of looks like a chicken to me, but this is an embryo at stages 5 to 6 weeks. And that is called the sexually indifferent stage. So here's how the Y chromosome influences the development of a fetus, right? So up to this point, week 5 or 6, the growing, developing fetus is at this sexually indifferent stage, meaning it's not determined yet whether this will become a male or a female. So sometime in that time period, the Y chromosome is going to start expressing this gene called the SRY gene. And the SRY gene is only found on the Y chromosome. I paused there because, of course, weird things can happen and it can hop onto the X chromosome sometimes. But normally, that SRY gene is only on the Y chromosome. So if you are female and you don't have the SRY gene, right, it can't get expressed. However, if you are a male and you have the Y chromosome and that SRY gene gets expressed, well, that is going to lead to the development of maleness. So in other words, the default development for humans is to be female unless that SRY gene is there and it gets expressed, then maleness will develop. So let's look at this picture a moment and you'll notice, can you find the gonadal ridge? Yes, great, right? So this gonadal ridge here, you'll notice there are two of them. Those will develop into the gonads. So if the SRY gene is turned on, those are going to develop into testes. If the SRY gene is not there, not turned on, those are going to develop into, nice work, ovaries. You'll also notice that there are these two ducts. So there's the mesonephric duct, also known as the Wolffian duct, and the paramesonephric duct, also known as the Mullerian duct. And that has to do with nephric, has to do with, nice job, nephrons. You'll see here, down here, the kidneys, the metanephros. The kidneys are kind of developing nearby this system. And if you think about it, urethras, kidneys, right? In one of the sexes, all those tubes converge together. In females, it does not. More on that in a moment. But so there are these two ducts, and if we have the SRY gene is present, then we're going to have a male, right? In that case, this mesonephric duct, this Wolffian duct, is going to develop, and the paramesonephric duct, the Mullerian duct, is going to degenerate, just disappears. The vice versa is true if we have a female developing. That Wolffian duct disappears, and the Mullerian duct, or the paramesonephric duct, is going to develop into the uterine tubes. Let's look at that. So here we have in just 7 to 8 weeks, right? So weeks 5 to 6, that gene gets turned on, and then we start developing the male and female reproductive systems. Sorry, that got a little squished there. So here we have 7 to 8 week male embryo on the left and an 8 to 9 week female fetus on the right. That's interesting. I just used the labels from your book. I hadn't noticed until this moment that one says embryo and one says fetus. I don't know what that means. Maybe nothing. Okay. So you can see here that in the males, that duct that's closer to the gonadal ridge, that is now going to become the testes, develops, the other duct disappears. The opposite is true in females. Now, that's an interesting thing anatomically to note, because for males, this tube is going to be continuous from the testes through the epididymis, through the vas deferens, through the urethra, in order to deliver the sperm and semen out of the body and potentially into a vagina. However, for the female reproductive system, the outer duct system develops and becomes the uterine tube. And what do you know about ovaries and uterine tubes? Yeah, well, I don't know how much you know, but here's what I can tell you is that never fuses, that the end of the fallopian tube hugs around the ovary, but it's not a continuous tube. That's one of the reasons why pregnancies can occur where the egg gets fertilized, but instead of getting catched by this end of the uterine tube, it bypasses it and it can implant on the outside of the uterus or the outside of the fallopian tube. It's one way an ectopic pregnancy can happen. As you can imagine, that's pretty dangerous for both the baby and mom. All right. And here we have, as things have further developed at birth. Fast forward in time. So you'll notice on the males, those gonads have descended outside of the body. Spermatogenesis happens much better at a lower temperature, a little bit around 93 degrees Fahrenheit instead of around the 98.6 degrees Fahrenheit that the body is. So having those gonads outside of the body helps with sperm development. Whereas the female gonads stay inside of the pelvis. And here you can see the uterine tubes have bent around, but there's still that gap between the fimbriae of the uterine tube and the ovary. Any questions? All right. So now that we've got this development of the male and reproductive system, let's get into gamete formation, right? Because that's what we need in order to make a new human. We use eggs and sperm. So here's one of the questions from your study guide, which is what stage of life does gamete formation begin and end for females versus males? And here's your clues. Extra points for anybody who recognizes our 13 to 14-year-old male. Yeah, that's the Biebs, Justin Bieber, at the age of 13, I think. So for females, the beginning of gamete formation starts in utero. So when that female fetus is still inside of her mother's womb, her ovaries are already starting this process of meiosis, which is the cellular division that leads to gametes, eggs, ova. That starts before she's born, and it gets stalled until puberty starts. Versus males, meiosis starts at the beginning of puberty, which is usually around 13 or 14 years old in most people. So, right, Bieber, he's still cool? That's okay. Still cool in my world. So what is meiosis? How is the process different for sperm and eggs? And why is it critical that gametes have only 23 chromosomes? Which question do you want to start with? All right, sure, start at the top. So what is meiosis? Well, there are two types of cellular division. There's meiosis and mitosis, right? If you ever have a hard time remembering which one, what I like is mitosis happens in my toes, right? It's the cellular division that happens that makes a whole body. Each one of those cells are genetic clones of each other. So remember we talked about this at the very beginning of the semester, that as you started to develop after fertilization and those cells start to divide through mitosis, each one of your cells is a genetic clone. What makes your cells look different from each other, do you remember? Oh, nice job. Yeah, you got there. Right. Yeah. So it's it's which genes are turned on versus turned off, right? So in your red blood cells, you have the hemoglobin gene is turned on, but of course that gene is turned off in cells like all of those cells. That's a good example. Your neurons, your skin cells, basically any cell that's not a red blood cell, that gene is turned off. Okay. So here's what's different with meiosis. Meiosis is going to produce gametes. So it's the only place that's happening in the body, ovaries and testes. And this type of cellular division does a few things. Most critically, it's going to reduce the number of chromosomes by half. So in most of your cells, all of your cells, except for your gametes, you have how many chromosomes? Oh, back it up 10 minutes, you'll find out. Okay, yeah, you remember, 46, right? 23 from the sperm, 23 from the eggs. Ah, right. So in order to half those number of chromosomes in half, meiosis goes through not one, but two processes of cellular division in order to reduce that number by half. And now you might be going like, wait a second, if I start with 46 and I go through one division, I get 23. Why do I have to go through 2 rounds of cellular division? Well, because in order to start cellular division, your DNA duplicates. Mitosis came first. So we're going to duplicate all of that DNA. So at the beginning of cellular division, I don't have 46, I have, 92. So I'm going to make a copy of each strand of DNA, the chromosomes. And so if I start with 96, I've got to go through 2 rounds of cellular division to get down to 23. All right, we're not going to go, you've worked through some of the details in mastering, and that's as far as we need to go. So you're going to go through these 2 rounds. So at the end, what I end up with are 4 cells, right? And the other thing to recognize about meiosis is each one of those cells is genetically unique. And that's pretty cool. That's why if you have siblings and you both have the same biological parents, you might have some similarities, but you look unique from each other, right? You're not exactly like your mom or your dad or like any of your siblings. That's because the sperm and the egg that join together to make you were genetically unique. And so every time a gamete is made through that process of some processes called crossing over and non-disjunction, actually, excuse me, crossing over and you got it, independent assortment. Nice job. Non-disjunction is something else. You get, unique gametes, genetic variation. We're going to skip down to the bottom one. So have you caught it? Why is it critical that gametes only have 23 chromosomes? Right. Humans have 46 chromosomes. If we didn't reduce the number of chromosomes in gametes, like if gametes were made through mitosis and they had 46 chromosomes, then when an egg with 46 chromosomes was fertilized by a sperm that has 46 chromosomes, how many chromosomes do I have now? 92, right? And that would happen with every generation. That's just, it makes a mess. It doesn't work. And so we need to make sure that those numbers of chromosomes have been halved. So you get half of your genetic information from each parent. So that's why it's critical. So let's focus on this one. How is the process different for sperm and eggs? Well, one thing you might be noticing on this chart right now, I'll give you a moment to look at it if you haven't yet, is, well, we start one's spermatogonium and one's oogonium. Okay, that's exciting. But notice at the bottom, I have 4 spermatozoa or 4 sperm that are functional, firm, that could, each one of those could functionally, one of those days. All right. Unfortunately, Zoom makes it so I can't stop and back it up and edit it. So we're coming at you live, recorded in front of a live audience of me and my microbiome. And I should stop rambling because we're going for it. Okay, so where were we? 4 sperm. 1, 2, 3, 4. So each one of these, presuming they're all healthy and happy, could fertilize an egg, right? It's different for meiosis with eggs. So the egg goes through meiosis 1 and meiosis 2. That's the first round of cellular division, second round of cellular division. But you'll notice I only end up with 1 ovum, meaning I only have 1 egg that's viable, that could be fertilized. The other ones become these polar bodies, and those are too small to be viable. So what's happening there is as the oocytes, so many terms, I know, but basically as the cells that are going to go through meiosis to form the egg, the ovum, as those cells divide, they don't divide evenly. Most of the cytoplasm goes into one cell, and that's going to become the cell that's going to become the egg. And you need a really big cell there because it's got a really big job to do. So that's one of the differences. We'll continue with differences as we go. But something that's kind of interesting to see is, do you have any idea, like 20-year-old male, healthy, how many sperm a day is that human making? Millions, yeah, about 90 million a day. It's a lot. Not as many red blood cells as you're making every day, but that's a lot of sperm. And of course, it's been somebody's job to look at that sperm underneath a microscope and categorize different types of sperm that get made, right? So there's the normal sperm, what we consider would probably be a healthy sperm and be able to fertilize an egg, make it through the long journey to get there. But there are lots of different types of defects that can occur during development of sperm. Here's some of them, which is kind of interesting to know about. That as that cellular division happens really fast, right? Not all sperm are developed correctly. And because that cellular division happens really fast, there's also metabolic waste products that are happening there that increase the chances of mutation. So if you're looking at mutation events, they're actually higher in sperm than they are in eggs. Trivia for you there. All right, so What starts puberty? I thought this one was fun. I'll let you read it. Yeah, puberty kind of feels that way, right? It's been a while, but yeah. Okay, so when does it start? Well, or what starts it? That was the question, not when. We got that. 13 to 14 years old. So what's happening there? Well, that has to do with a hormone feedback loop. Of course it does. So here are the players involved. Remember the master key switch of your endocrine system, the hypothalamus and the pituitary gland. And now we're hanging out with the anterior lobe of the pituitary gland, and then the gonads, right? So testes in men, ovaries in women. And those are going to produce sex hormones, right? Testosterone and estrogen mainly. And how does this feedback loop work? Well, I'll give you a moment to look at it. The dashed lines mean, have you seen that? Inhibits, right? So here we have a negative feedback loop. So let's start up at the hypothalamus. So the hypothalamus is going to release a hormone known as GnRH, gonadotropin releasing hormone, right? The hormone that is going to release something that impacts the gonads, and tropin just means one hormone affects another. So that gets released from the hypothalamus, and that stimulates or promotes the release of LH and FSH from your anterior pituitary gland. LH stands for luteinizing hormone. That's named after the female component. We'll talk about that later. And component, I don't know. And FSH, follicle stimulating hormone, also a, we'll go with it, component of the female reproductive system. So we'll show you those a bit later. But both of those, even though they're named after their roles in the female reproductive system, both males and females make GnRH and LH and FSH. And both in males and females, the LH and FSH, there are receptors in the gonads that cause either the testes or ovaries to make more sex hormones, right? To make more testosterone in the testes and more estrogen in the ovaries. It's more complicated than that, but that's what we're sticking with at the moment. It also makes this hormone called inhibin. So you'll notice that those sex hormones have target cells, and those target cells are, well, there's a variety of them, but one of them is the cells that are going to make the sperm or the eggs, right? That's going to promote the formation of gametes. There are also cells that are going to receive those hormones that develop our secondary sex characteristics, right? All right. So back to our feedback loop. So as those sex hormones increase, so things like testosterone and estrogen, there's this negative feedback loop, meaning as testosterone increases, what's going to happen to my GnRH levels, my gonadotropin releasing hormone levels? All right, it's going to decrease. Same thing with my LH and FSH, right? As those increase, then those meaning testosterone and estrogen, it's going to cause this feedback loop and reduce the levels of the GnRH, LH, and FSH, right? Negative feedback loop. Okay, you've got it. So what does that have to do with initiating puberty? Well, at puberty, the hypothalamus, oh, there should be an S there. I'm going to fix that right now. Wait for it. All right, I'm back. You didn't go anywhere. So one thing to recognize is that the hypothalamus and the anterior pituitary gland and the gonads are going to be releasing low levels of these hormones throughout your life before puberty. And actually there's a surge of testosterone early on in development for males. You can look at that in your book. But the hypothalamus, right? Even though there's these low levels, thank you, Erin, and this feedback loop is going on. So at puberty, we get this surge or this increase of levels of sex hormones, right? Testosterone increases, estrogen increases. That's what starts puberty. That's what allows for the beginning of the development of gametes, spermatogenesis. It's going to continue oogenesis, egg development in females. We're going to get the secondary sex characteristics. Females are going to start menstruating. So all of that comes about because we have this increase in the sex hormones. Well, how does that happen? Well, what's going to increase your levels of sex hormones? LH and FSH. What's going to increase that? GnRH. Well, what's going to increase your GnRH levels? Oh, that's interesting. Because if I'm increasing my levels of sex hormones, it's a negative feedback loop, right? So the rest of those should be decreasing. So something has to change in the hypothalamus, and it does. Around the age of puberty, the hypothalamus becomes, what do you think, more or less sensitive to those sex hormones? What's going to let the hypothalamus release more GnRH that's going to lead to more LH and FSH and more sex hormones? Are you sure? Yeah. All right. It's going to become less sensitive to it. Does that make sense? So if your hypothalamus is becoming less sensitive to sex hormones, think about it like if it has fewer receptors. Oh, wait, forget that. Let's just stick with this. Let's not get into the molecular pieces of it. So if your hypothalamus becomes less sensitive to the hormones, right, then I'm going to need more hormones for that feedback loop to have an effect. The hormones are going to have to yell louder at the hypothalamus and the anterior pituitary gland in order for them to have to inhibit the release of GnRH or LH and FSH. And it's specifically the hypothalamus. So if the hypothalamus becomes less sensitive, then those sex hormone levels can rise before it starts releasing GnRH. And that's how it works. And so slowly over puberty, right, over that 3 to 4 years that puberty is happening, the hypothalamus is changing its sensitivity. It's becoming less and less sensitive until you get to this point where we're reaching this equilibrium between the sex hormones, LH and FSH, and GnRH. Well, sort of. They also, for women, have this 28-day cycle, but they work it out. So it's not continually increasing. Okay. Then let's compare and contrast the roles of GnRH, LH, FSH, and sex hormones in spermatogenesis and oogenesis. And surprise, I realized I missed something that was in the study guide. So I'm going back to it. So one of the questions is, how is the process different for sperm and eggs? And we talked about how the numbers are different. And we talked about how meiosis starts at puberty for males and it starts before birth for females. But there are a few more details I want you to recognize, right? So if it starts before birth for females, then what happens? Like what's going on at puberty? So this primary oocyte, right, the beginning of meiosis, is developing before birth. And then the secondary oocyte here, so we were starting meiosis 2, is what gets started during puberty. So basically, these cells are kind of on hold. They call it arrested from the point of before birth to puberty. And then at puberty, meiosis gets started again as follicles start to develop. Follicles are the cells around the egg, right? So as the egg starts to mature and the follicles around it that support it start to mature, then meiosis starts again. And meiosis 2, so the end of meiosis 2 only completes if the egg gets fertilized. Yeah. So meiosis in men from start to finish takes about 60 days. In women, it can take 40 years, 30, 40, 20 years, 15 years, depending on when that egg develops, right? So not all eggs are developing at the same time. So every month, a few oocytes start to develop, around 20 or so. The largest one is the one that becomes ovulated. The rest of them atrophy, they die off. And the largest one, the most dominant one, is the one that gets ovulated. Occasionally, 2 of them will be released, and then you can get fraternal twins. Sorry for the jump back, jump forward. We're going to jump forward again. You ready for our time hop? So back to the hormonal feedback loops and development of spermatogenesis and oogenesis. So we're going to start with spermatogenesis, which is a little less complicated, and then we'll get into oogenesis, which is a little more complicated. But it's all based on this basic feedback loop between the hypothalamus, the anterior pituitary gland, and the gonads. It's true for both sexes. So in the male reproductive system, here, recognize that sperm are being made in the testes. That's where spermatogenesis occurs. They're not mature yet. They're still immobile. So they're going to spend time in this other tube here, which is known as the epididymis, until an ejaculatory event, at which case they'll follow the vas deferens up over the pubic bone, around the back of the bladder, through this what's called the seminal vesicle, which helps produce the semen, and then out the urethra. It's a long trip. It's that long trip because, remember, these started up here as those gonadal ridges, and then during development, those testicles descend out of the body. Okay, so here we are inside of the testicle, which is full of tubes. Those tubes are called seminal tubules. Here's an electron micrograph. You can see the tails of the sperm, which is to just show you that these tubes have stem cells that are towards the outside of that tube. As those stem cells divide through meiosis, then they're going to be pushed forward into the center of this tube. Remember, they're not mature yet. They actually can't swim yet. And so they are through acts of peristalsis. There are special cells called myoid cells that can contract those tubules and move those sperm into the epididymis where they can mature. So I just want you to notice here that there are, we're going through meiosis and we're ending up with these 4 sperm that are genetically, nice job, unique. Okay, so let's talk about the hormonal feedback loop. All right, let's walk through it. So we're going to start at the top with the GnRH that's getting released from the hypothalamus. That's going to promote the release of FSH and LH, the follicle stimulating hormone and the luteinizing hormone. All right, so here's where they do some different jobs. So luteinizing hormone is going to have receptors on some cells called the interstitial cells. Where are those interstitial cells? Well, you'll notice they're outside of these tubes. So if we go back up here, if this is our seminiferous tubule, you'll notice there are some cells outside of those tubes. So that's where our interstitial cells are. These are the cells that have receptors for LH. And LH tells these interstitial cells to produce testosterone. So the sperm themselves do not make testosterone, rather it's these interstitial cells. And those interstitial cells, when they're producing testosterone, well, then that testosterone can then be released into the blood, and it's going to have somatic and physiological, psychological effects on other body sites, right? It's going to do things like increase muscular mass during puberty, changes the voice box during puberty. It's also responsible for libido in males. And back to our sperm development, FSH, that follicular stimulating hormone, is going to talk to these sustentacular cell. It's sustentacular. So the sperm, as they're developing, are supported by these larger cells that they're kind of embedded in. And those cells can release something called ABP, which of course is short for androgen binding protein. And testosterone is an androgen. So basically what this does is it binds to the testosterone and keeps concentration levels of testosterone close to the stem cells that are going through meiosis to develop sperm. So you need testosterone in order for those stem cells to go through meiosis. So it's just keeping the testosterone closer. Voila. So that's how those two hormones work together to produce sperm. And of course, during that process of developing testosterone and developing sperm, the testosterone itself acts as a negative feedback loop to both the anterior pituitary gland and the hypothalamus. And another hormone gets released from that seminiferous tubule from the sustentacular cell called inhibin, and that's going to inhibit FSH and LH from being released from the anterior pituitary gland. As I said, it kind of balances out and they get this feedback loop going during puberty until you get to the point where you can make 90 million sperm a day. So that's the feedback loop for spermatogenesis. Let's talk about the feedback loops for oogenesis. So here we have the female reproductive system, where we have the ovaries, which are going to produce the egg, also known as the ovum. A premature ovum is also known as an oocyte. It's not an oocyte, it's pronounced oocyte. I guess you can say oocyte if you want to. And if that ovum is fertilized, then it's going to travel through the uterine tube regardless, but fertilization happens in the uterine tube. And it takes about a week for it to travel into the uterus. If it's fertilized and the uterus has developed an endometrium that can support a pregnancy, then it can embed and pregnancy can ensue. If that ovum egg is not fertilized, then it dies within 24 hours of ovulation. So even before it gets through the uterine tube, that cell has died, and that cell is just going to get sloughed off when a woman menstruates. Just me checking to make sure. Oh, so what you're looking at here is in this ovary, each one of these larger cells here with the purple center would be a developing oocyte, which will become the mature ovum, and they've started meiosis as primary oocytes. These are these small ones here on the edge. As they develop and mature, the oocyte becomes bigger. And you'll notice it also gains a bunch of supportive cells, and those are called the follicle cells. So as the cell's growing larger and it's going through, it's completing meiosis one. So meiosis one started in utero, right? Stalls out until puberty. And then a few of those oocytes are going to start developing every month, which is why if we look at a microscopic view of an ovary, you can see at any given time, there's more than one maturing oocyte. And as it develops, eventually it's going to develop this fluid around the developing oocyte and its follicles. And a few more and more of these cells are going to die off. That helps separate that oocyte. And then when it leaves, we call that moment ovulation. Now, is meiosis done yet? No, correct. It is paused at the beginning of the second round of cellular division, meiosis 2. When will that finish? Yeah, if it gets fertilized. Okay, so you ready for some feedback loops? Let's do this. I know, it's just the beginning. Okay, so things are a little bit different and a little bit the same of the male reproductive system. So let's walk through it. So we have this hypothalamus is releasing the GnRH to the anterior pituitary gland. That's going to release LH and FSH. The LH and FSH, as you can see by those targets in the ovaries, have slightly different targets. And they have, just like they did in the male reproductive system, right? Both of those are going to work together to help meiosis happen and help that cell mature. So essentially, the key piece here is both LH and FSH are going to increase the amount of estrogen that's being made. Slightly different targets, slightly different pathways, but they're both increasing the estrogen. And that's going to do a couple of things, of course. So the estrogen, just like testosterone in the male reproductive system, we're going to follow this dashed line, and it's going to cause this feedback loop, and that's going to cause the GnRH and the FSH to decline a bit. Also, with that, inhibin is playing the same role. That's going to inhibit the development of, or the release, excuse me, of GnRH and FSH. Okay. Which, if you look at this other diagram in your book, this is fun, you can see here this is the area we're in right now, is the development of the ovary, right? We're going from these primary follicles to one dominant follicle, right? Talked about how it's more than one follicle or oocyte that starts to continue going through meiosis at the beginning of this ovarian cycle. Happens every 28 days. So as we have more of these oocytes being developed, right, we're having more estrogen. So we can see the estrogen is starting to come up. So as that estrogen is increasing, then our FSH is declining a bit. So that decline in follicular stimulating hormone, that's where it's got its name. Without that support of having that hormone, most of the other follicles are going to die off. And the largest one, the dominant follicle, is large enough that it's still getting signals from the FSH. And as it continues to grow, it is going to produce more estrogen. And there's a moment where this dominant follicle is going to have this surge of estrogen and release it much higher than the previous slow increases of estrogen. And that surge changes the game. So that surge, now we're down here, here's our mature follicle. It's going to have that surge of estrogen that gets released from these follicle cells that are developing. Note, the egg itself isn't producing estrogen, just like the sperm is not producing testosterone. So the gametes aren't producing the hormones, it's the supporting cells. So we get the surge of estrogen, and that, ooh, look, it's a solid line. That's causing a positive feedback loop. I promised you it gets complicated. So that causes this positive feedback loop. So now, instead of decreasing the amount of GnRH, we are increasing it. Same with the anterior pituitary gland. Now we're going to increase the amount of LH and FSH that's being released from the anterior pituitary gland. So you'll notice 5 says LH surge. And if we look at this other, right, we have an increase in both the FSH and the LH, but clearly the LH is a bigger surge. So the effect of that positive feedback loop of an increase of the spike in estrogen is that we have this spike in luteinizing hormone. And what the luteinizing hormone does, I don't know why they didn't call it ovulating hormone. I wasn't in charge, but it causes ovulation. That signal of luteinizing hormone causes the ovulation. Woo, wrong direction. This secondary oocyte, which is called secondary because it's in the second stage of meiosis 2, gets released. All goes well. It gets caught and swept up by the fimbriae, the end of our uterine tube, enters the uterine tube where it can be fertilized if sperm are present. If not, it dies within 24 hours. But it's that surge in what hormone? Did you say it out loud? Did you write it down? Did you teach your cat? Okay. Or your dog. A lot of you have dogs. That's great. Pets are great. Just gonna stay focused. So what's this thing stand for? Luteinizing hormone. What the heck is that? Meet the corpus luteum. Luteum means yellow. Corpus means body. It's the yellow body. It makes a yellow pigment. Yeah, I don't know. I don't know what makes the yellow pigment, but I do know it's yellow, and that's how it got its name. Where did it come from? Well, you'll notice not all of the follicular cells leave with the oocyte, the ovum that's been ovulated. There are a lot of those follicles that remain. And remember, it's the follicles that are making most of the estrogen and progesterone. We haven't talked about that one yet, but it's in there too. And so a lot of that remains, and after ovulation, those cells form this thing called the corpus luteum. Great, right? That's what is being shown here. Ovulation, these cells that were supporting the development of our dominant oocyte, a lot of those stay, it becomes this corpus luteum. Okay. The corpus luteum, which was these follicle cells, continues to produce hormones, progesterone, estrogen, and inhibin. Estrogens. It's plural because there, of course, there are different types of estrogen. I'm not going to get into that. But we see elevated levels higher than before of progesterone, estrogen, and inhibin, and that's going to dashed line. So we are inhibiting the release of gonadal GnRH, LH, and FSH. And that's going to continue, which should make you pause and think, well, then how's the next egg going to develop? What's about our next round of oocytes? Well, here we go. So let's focus on this ovarian phase here. So you have these follicular phases, right? We've had the ovulation occur. These cells here have become the corpus luteum. It grows for a moment as it does that, it's increasing the levels of progesterone and estrogen, and that is going to cause feedback on our LH and FSH and keep those lower. Now, if fertilization happens and that egg becomes embedded in the uterus, then a pregnancy occurs. That there's another hormonal feedback loop that goes between the uterus, the growing placenta, and the ovaries, and that corpus luteum stays. And those progesterone levels and those estrogen levels stay elevated throughout the pregnancy because estrogen and progesterone are being continued to be released by that corpus luteum, and the uterus itself also starts to produce some hormones, and that suppresses LH and FSH, and so that's going to prevent further ovulation during the pregnancy, which kind of makes sense, right? You don't want to have one fetus that's like 4 months along and then get pregnant with another fetus. Would get complicated. I know kangaroos can do it. It's pretty cool. We can talk about that later if you want to, but humans don't have that superpower. Okay. So if the egg does not get fertilized, then, and there's not that feedback between the uterus and the corpus luteum, then the corpus luteum degenerates. Aha. So when the corpus luteum degenerates, then it stops making, right? We're going to decrease down here in the plasma hormonal level chart. We're decreasing the amount of progesterone and the amount of estrogen. And as we decrease that, we're not, yes, and the inhibin, all of those are decreasing. And so that's going to allow the, nice job, LH and FSH to start to, good job, increase. And that's going to start this whole ovarian cycle over again, where we can get a bunch of follicles developing, one becomes dominant, et cetera, et cetera. Pretty fun, right? So the male reproductive system stays a negative feedback loop between the sex hormones and the GnRH, the FSH, and the LH. Females are different in that most of the time it's negative, except when we have this, you remember the exception? Yeah, this big spike in estrogen, that big spike in estrogen that's caused by this dominant follicle that's gotten large enough that those cells are just going to do this big surge of estrogen. That is a positive feedback loop that causes the LH to surge, nice work, and that LH surge is what causes ovulation. Okay. So while all those cycles are going around, in the ovary, we also have the uterine cycle. So the uterine cycle starts at the same time as the beginning of primary oocyte development. So when the primary oocytes are just starting to re-enter meiosis, that's when the menstrual flow is happening. So menstrual flow is happening, the endometrial layer that's leaving, that's being shed through the vagina, that happens for 3 to 5, 7 days, depending on who the human is. And then that lining starts to build back up again. During that uterine phase, it's responding to hormones. It's so that it's ready by the time the luteinizing hormone does its spike, and ovum is released. Do you remember how long it takes for that journey to happen? Yeah, about a week, right? So it takes about a week for that egg, if it gets fertilized, to get into the uterus, at which time the uterus should be ready to receive that fertilized egg and allow pregnancy to happen. Voila. Let me know if you've got questions. So what about birth control? So there are different types of birth control, right? Some of them are more anatomical in that they're just blocking the sperm and the egg from meeting, whether it's a condom or a diaphragm. But since this is physiology class, I thought we should talk about physiological birth controls. So one of the more common types of birth control are the pill and IUD devices. And of course, there are other types. If you're interested in those, I could encourage you to go check them out. But the pill is either just progestin or a mixture of progestin and estrogen. And now, can you see why that works? Aha. If I keep progestin and estrogen elevated, I'm going to stop ovulation. We'll get to the reduce in a second. So if I have a pill with both of those and both of those stay elevated, having both of them elevated does almost 100%, 99% job of preventing ovulation from happening, because these are high, so it's a negative feedback loop. So I am preventing, right, the LH and the FSH from secreting, and so I'm preventing meiosis. I'm preventing the development of these primary oocytes into secondary oocytes into a fertilizable egg that has ovulated. So we're going to prevent ovulation because we're preventing meiosis. Some pills only have progestin, and progestin is a synthetically made version of progesterone. Those pills might reduce ovulation, but one of the primary ways they work is there's in the cervix, the body makes really thick mucus. It's called the cervical plug or the mucal plug. And that serves as a physical barrier for the sperm. So one of the other things that's happening during this 28-day cycle is near the time of ovulation, that mucus that's in the cervix changes and it reduces in thickness. So it's easier for the sperm to swim through it. So a progestin pill is, they think it probably reduces ovulation, but it's also really thickening that mucus layer. But it's not always effective as the pill that has the progestin and the estrogen, but some women having the pill with both, the estrogen causes side effects that are not fun. The other form of birth control, or another form, there are lots of forms, is an IUD, an intrauterine device. So some of them are hormonal, some of them are copper-based, but either way, that is inserted into the uterus and can be good, depending on the type, is good for 3 to multiple years, depending on the type you've got. So the hormonal releases different hormones, progestin, estrogen, and it's it's also going to thicken the cervical mucus, and it's thought to reduce sperm transport. They're not really sure how, other than it thickens the cervical mucus, but even then it seems to reduce the ability of the sperm to get to the end of the uterine tube. It also, for a lot of women, will thin the endometrium. And so they don't build up a thick enough endometrium to support a fertilized egg. And it may suppress ovulation, but that's not always clear, or it varies from woman to woman, whether or not it suppresses ovulation. But since it does thin that endometrium, even if the egg is fertilized, implantation won't happen because there's really no, the endometrium isn't thick enough to support a pregnancy. The copper seems to work because sperm don't like copper. So it inhibits their ability to swim, or if they can swim, they avoid the copper, so they don't swim in the right direction. And that also seems to change the uterus in a way that it can't support an embryo. And so it prevents implantation if in the event that ovulation and fertilization do happen. All right, let me know if you got questions. Male birth control. So these are still in research, but there are actually quite a few lines of research for male physiological birth control, which has not been available to men or couples so far. But this came out March 25th, 2022, male birth control pill expected to start human trials this year. So it's been effective in mice as the model. In mice, it prevented pregnancy for 99% of their mice couples. And they're hoping to start it in trials, just like I said. So let's talk about those a bit. So there are lots of different types of male birth controls that are on currently in development. So some of the earliest ones were seeking to reduce testosterone levels. So you'll remember that in order for spermatogenesis to happen, you need to have testosterone. So some of the early developments were hormonal pills that were working to reduce testosterone levels. Those had all kinds of side effects that could range from increased LDL to a decreased libido, to decrease in bone density, because testosterone helps increase bone density in men. So they've worked on developing a different non-hormonal pill that is called DMAU. Ooh, and I forgot to, hold on, let me get my acronym. Of course, DMAU, which of course stands for dimethylandroline unidecanoate. I can't see any reason why they decided to just call it DMAU. Anyway, when you do your Labster, your online lab this week, you'll be working with rabbits and DMAU as a way to reduce fertility in rabbits in a petting zoo. You'll have fun with that. So DMAU acts as a testosterone mimic, but it doesn't have the side effects. So it seems to suppress FSH and LH and suppress the development of sperm without possibly having the side effects of reducing testosterone. So it's going to start possibly, it's also in clinical trials right now, but it hasn't hit the market. There are also other labs that are looking at trying to decrease sperm motility, and that would have a more immediate effect. So anyway, and it's thought that that could potentially be used by both sexes, that whether the sperm are developing in the males or inside of the female reproductive system, that if you had that pill available to you, you could stop the sperm from swimming and then you would prevent pregnancy. And then the other one is preventing fertilization, and that targets the acrosome. So the acrosome is, did we talk about this? I can't remember. You can push fast forward if we talked about this, but so when they're looking at decreasing sperm motility, they're dealing mostly with the mitochondria here and the tail, right? So something that's going to reduce their ability to swim, or sometimes they're trying to get them to just swim in circles, so they never get anywhere. And then to prevent fertilization, the very tip of the head of the sperm, right? So in here, we have the nucleus that has those 23 chromosomes in it. And then we have this portion called the acrosome. And the acrosome contains a bunch of enzymes that help the sperm penetrate the egg. So there's this layer around the egg that has to be degraded through enzymes in order for the sperm to conduct fertilization. So some studies and research labs are trying to prevent the acrosome from being developed on the sperm, which you can't have fertilization if you don't have an acrosome. And then this thing that we just looked at, the one that's going to trial next year, vitamin A is required for sperm development, and there are specific proteins that bind to vitamin A during sperm development. And so this particular pill is blocking those proteins that bind to vitamin A during sperm development. And there, at least in the mice, there seem to be no side effects. So it's going to enter clinical trial. Voila. We've made it to the end. So thanks for a great semester. Remember, on so much of this, we've just scratched the surface on the beginning of physiology and how the human body works. So hopefully your curiosity has been engaged throughout the semester, and hopefully after the semester, as you start to get curious about how does that work in the human body, you've got a nice foundation now to work with as you explore those questions. It's been a pleasure. Thanks for being part of class. See you around.